The endosymbiotic theory is a theory that eukaryotic cells evolved from prokaryotic cells through a symbiotic relationship between some prokaryotic cells. In easier terms, prokaryotes are what we know every day as bacteria. There will be several bacteria that we will talk about: the cyanobacteria, aerobic bacteria, and large bacteria. The cyanobacteria is a bacterium that gets its energy from photosynthesis. Does this sound familiar? The chloroplasts of our eukaryotic plant cells do have the same properties of carrying out the process of photosynthesis. The aerobic bacteria refers to all bacteria that relies on air for survival, as it comes from the word aerobic respiration. Aerobic bacteria can produce energy known as ATP, which is useful in the cells. Their properties are similar to those of a mitochondria. Lastly, the large bacteria are bacteria that consume other prokaryotic cells that are also bacteria. This could sometimes be called a predatory cell, coming from the word predators eating their prey. When a blob-like prokaryote absorbs another prokaryote, sometimes this bacteria may have different properties, such as if it produces energy like a mitochondria, forming a eukaryotic animal cell, as all prokaryotes do not have a mitochondria.
Later, if the eukaryotic animal cell consumes cyanobacteria, it will become a eukaryotic plant cell. This is because the cyanobacteria eventually becomes chloroplasts. Now, you may be wondering, where's the evidence to support this theory? Well, here are some of the evidence.
Mitochondria and chloroplasts have their own DNA. It is separate from the DNA that's found in the cell's nucleus. Both mitochondria and chloroplasts still have similar ribosomes and a circular single-strand DNA to bacteria.
Another piece of evidence to consider is that cyanobacteria and aerobic bacteria and bacteria are similar in size. The last piece of evidence is that the ways cyanobacteria and aerobic bacteria and bacteria divide or duplicate themselves are similar to each other.
Mitochondrial DNA can be used to trace the geographical distribution of genetic variation for the investigation of expansion, immigrations, and other patterns of gene flow. And it can also be used when samples such as teeth, bones, and hair without follicles, the roots, are collected from crime scenes or disaster areas. Because mitochondrial DNA has a unique pattern of inheritance, it is passed down directly from mother to child. Unlike the nuclear DNA, which is passed down from both parents, it also accumulates changes more slowly than other types of DNA.